Since yesterday, I'm suffering from a lot of hiccups frequently, and I couldn't stop this morning, so hopefully it does not come back this afternoon, okay, so that we can finish our lecture. All right, we'll finish as soon as possible. I know it's getting cold and it's like winter now, and today we'll talk about hepatitis viruses, and we'll introduce all the known hepatitis viruses ever discovered by now. Okay, in this name, you know the meaning of hepa? Liver. Hepatitis? Exactly, so liver inflammation. So hepatitis virus is a group of viruses which can cause liver infection, inflammation, and liver damage. All right, so remote is not working. Okay, we'll introduce 5 different hepatitis viruses. The name is easy to remember: hepatitis A virus, B virus, C, D, E virus, short for HAV, HBV, HCV, HDV, and HEV. Okay, 5 different viruses. And okay, this is a summary table of all the 5 viruses. Okay, 5 different viruses, A to E, and it includes all the information of the major characteristics, such as the family they belong, genus, the variant size and symmetry, whether they have envelope, their genome property, genome size, stability transmission, or until whether they can cause liver cancer in humans. So it includes all the information we want to talk about today in detail. So I'd like you to utilize this table to compare the 5 different viruses so that you can master the key points, knowledge related to hepatitis viruses. Give me a minute. Let me change the remote. Sorry. Yeah. Okay. Okay, the first one we want to talk about is hepatitis A virus. Always takes time to settle down. Who is playing games here? All right, the first type of virus is HAV virus. We haven't heard of HAV outbreak in China for a long time, especially the like large scale of HAV infection. There might be some isolated HAV infection cases. This is what happened in last century. Happened in 1988 in Shanghai city. You know that Shanghai is one of the biggest cities in China, and there was an HAV infection outbreak in Shanghai in 1988. Because of utilization of this shell, it's a seafood, and Chinese name here is maohan, this type of seafood. And for some reason, the seafood got contaminated by HAV. And we know that HAV virus is relatively stable to heat. And if it's not well completely cooked, thoroughly cooked, you cannot completely inactivate the virus. So that year, a lot of people got infected in Shanghai city. And you can see that the hospital is crowded with a lot of patients coming into the hospital. And as I know, that's the last outbreak of HAV infection in China. So here's some information on HAV. HAV virus belongs to hepatovirus, only infect the liver. Structure of HAV, you can see here. So it's roughly spherical structure. Because of the cubic symmetry, it's actually from the icosahedral symmetry. So it's spherical under electron microscope. The genome of HAV is linear single-stranded RNA. Linear single-stranded RNA is positive sense. Size of the genome is 7.5 kb. And it's a naked virus, so there's no envelope covering the spherical. HAV is stable to treatment with ether, acid, and heat. And this is the common properties of the naked viruses. Because of the lack of envelope, the proteins are relatively more stable. So they are stable to treatment with ether, acid, and heat. And there are several primate cell lines which can support the growth of HAV. Primate cell lines. But HAV, when it infects the cell line in vitro, like in a lab, it does not cause any cytopathic effects. And this is kind of not regular for the naked virus, because usually naked virus infection can cause cytopathic effects we mentioned before, but not for HAV, even though it's a naked virus. I think the major reason is that HAV virus replicates gradually, slowly in the cell, so it does not kill the whole cell. Okay, the HAV virus. This is the genome of HAV, and total size 7.5 kb, single-stranded positive sense RNA. You don't have to remember the detail of the genome structure, but we should know that on the 5' end here, it encodes mainly the structural protein. So the structural protein, like the procapsid and the protein of the virion, the structural protein on the virion. Well, on the 3' end, it mainly encodes the enzymes, the non-structural protein, including the helicase, protease, and polymerase. So that's the non-structural proteins. Transmission of HAV is through fecal-oral route, like contaminated food or water. If ingested, it can set up infection. Incubation time is about 4 weeks. About 4 weeks. But one thing we should know that the person who is infected by HAV, even at the time of incubation, so before the onset of disease, they have already released a large amount of virion particles. So they release a lot of large quantities of viruses during the incubation period. That makes it hard to prevent HAV transmission if there are a lot of infection in some area, some city. So we should work on the environment, like treat the environment, inactivate the human waste before it's released. Clinical findings of HAV infection include subclinical, so that's the infection without any disease, without any symptoms. But it can also cause hepatitis, both like ecteric hepatitis and anecteric hepatitis. Here, this word, ecteric means jaundice. You know jaundice, right? You learn that. That's because of the liver cell damage that the metabolism is interrupted so that the patient's skin would turn yellowish color. So that's the jaundice. And right now, there is no evidence for progression to chronic liver damage. So that's a good thing of HAV. So after infection, most of the patients would recover, and it does not cause a chronic disease. And that's different from HBV and HCV. We'll introduce in detail later. Okay. So this is the the different phase, different time after HAV exposure. Time 0 is the exposure time, and the symptoms starts here, 4 weeks. Okay, so from time 0 to 4 weeks, that's the incubation time, incubation period. But if you check the virus load either in the patient's blood or in the patient's feces, you could see that in the patient's blood and the feces, the virus load peaks at about around 2 weeks. Okay, so that's before you even notice a person is infected. So it's not easy to prevent the spreading of the virus. While the symptoms comes at about week 4, and aminotransferases also peaks at about week 4. You know the source of aminotransferases? I guess we learned that in biochemistry. Aminotransferases is originally from liver cells, hepatocytes. It's intracellular enzyme. Okay, you shouldn't detect any of these enzymes in the serum if the cell is healthy, is intact. Only when the liver cell is damaged, these enzymes can be released. So the presence of aminotransferases in the serum indicates the liver damage. The liver cell is damaged. Okay, this is what happened at different times. While if you check the antibody in the serum, this is the IgM, which increased first, then declines. While IgG, it will increase at about week 4, then it will last for a long time. That's HAV. And based on this, based on different time points, you can decide which specimen you want to pick for examination. Okay, prevention and control of HAV. We should interrupt the transmission route. We know that the HAV transmits through the fecal-oral route, which we should try to interrupt the transmission route. Like from the fecal part, we should treat that, inactivate the human waste, so that we can inactivate viruses in the waste, in the feces, before it's released to the environment. And we should improve the hygiene condition in the environment. And before we use any possible contaminated food or water, we should like cook it thoroughly or boil it thoroughly to kill the viruses in it. And we can also use immunizations to prevent HAV infection, both passive immunization and active immunization. Passive immunization, we use immunoglobulin, so the pre-existing antibody, to neutralize, to bind to the HAV virus. So that can prevent 80 to 90% of any possible infection, prevent 80 to 90% in preventing clinical disease symptoms. So that's a passive immunization. After exposure, we use antibody to block the infection. Or we can use active immunization. You know the meaning of active immunization? So we use antigen, the vaccine, to stimulate our own immune system. Right now, there are two types of HAV vaccine available. That depends on different countries. Like in China, right now we have both the killed HAV vaccine and also the attenuated live HAV vaccine available. The killed HAV vaccine is included in the vaccination plan, which is free, while the attenuated live vaccine is not free, I think. Do you know what type of vaccine you received when you were little? No? Probably you received both A and B. Okay, okay. We should have a record either online or on a paper, including all the information. So this is the active immunization. When I was little, I think previously we only have the killed, inactivated HAV vaccine. And as I understand, the inactivated HAV vaccine is effective enough because it actually inhibited HAV infection in China. And there are no specific treatments for hepatitis A. So for those patients who are already infected, there is no specific medicine available, but you can support the patient to recover. So this is the first virus, HAV. While for hepatitis B virus, HBV, it is belong to the genus HepDNA virus. HepDNA virus. Heper is liver. DNA, that's the genome of its the genetic material is DNA. So in this genus, there is only one type of virus, that's HBV. And if you check the specimen with electron microscope, you would find 3 different morphologies of HBV. Some are small spherical structures like this, small spheres. Some are filamentous or tubular structure. While you can also find occasionally some big particles like this. And those are different particles, they contain different material. For the first one, the small spherical particle, the small one, the diameter is about 22 nanometer. The only chemical component of this particle is the HBV surface antigen, one type of protein, surface antigen. Okay, this is the first type. The second type is a tubular one, this one, or filamentous particle. The diameter or the weight is I'm glad you are learning Chinese. Okay, the filamentous one, the diameter here is again 22 nanometer, and the material in it is only the surface antigen. Okay, but the filamentous one, it can be very long. It's like 200 nanometer long, so that you can see this special structure like this. Okay, the filamentous structure. Both of them only contain the surface antigen. So those particles cannot infect a person because it does not contain any nucleic acid. It only contains a simple protein. While another particle, the DEM particle, is the infectious one. So this is a complete HBV variant, which contains the complete structure of the viral particle and also contains the genome in the middle. The DEN particle is named by the scientist who discovered the, the, the particle. The diameter here is 42 nanometer, so much bigger than the small one. But it's infectious because this is the complete HBV variant particle. But it is less frequently observed in this specimen. And if you dissect the particle, you would find the material, the structure of the particle here. Okay, the total diameter is about 42 nanometers. It has envelope, the outer layer, that's the envelope. On the envelope, it contains a lot of protein, especially those surface antigen. Okay, there are 3 different types of surface antigen with different size, protein size, large, medium, small, okay, on the surface of the envelope. And even though it has an envelope, but this envelope is special that it carries a lot of protein molecule and less lipid molecule, so that this envelope is more resistant to ethanol. Okay, and in the middle, that's a nuclear capsid. Okay, a protein shell with the genome inside. And on this capsid, you would find the core protein, core antigen, the C antigen, okay? And it carries DNA polymerase in a variant particle. And its genome is DNA, HBV DNA. HBV DNA is a very special DNA that it's like, from this picture, it's like a circular DNA, right? It's like a plasmid. But actually, the DNA is not a complete double-stranded circular DNA. It's a partially double-stranded circular DNA. And you can see here, the most part of it is double-stranded. But for this part, it's only single-stranded. We call it partially double-stranded circular DNA. Size of the genome is about 3.2 kb, 3,200 base pair. Much smaller than HAV, right? HAV is 7.5. And on the genome, there are 4 open reading frames. The first one is open reading frame S here. it encodes 3 antigens. The major one, the surface protein S, HBV surface antigen, but it also encodes the pre-S1 and pre-S2 antigen. That's the surface antigens. And in the open reading frame C, the core open reading frame here, it encodes the C antigen. And this C antigen can be further processed to make the E antigen. So both C and E antigen is encoded in the same open reading frame. And these 2 antigens, the C antigen is in the middle of the structure. So if you detect the presence of C antigen or E antigen, it means that the virus is replicating. So there is active replication of HBV. Well, another open reading frame is P, it's here. It encodes the polymerase. This HBV polymerase is very special because it can carry out different functions. It can work as a DNA polymerase. It can also work as reverse transcriptase and RNase H. And these activities are required for the life cycle of HBV. Later, let me introduce the life cycle of HBV. And the last open reading frame is the X, this one. That's a transcriptional transactivator. And this X open reading frame, the product of this one, the protein, is the major reason of transformation. So that after HBV infection, it can lead the transformation of human cell to cause liver cancer. Let me show you the life cycle of HBV. It's the replication of HBV. HBV is a DNA virus. We learned that before. That for DNA replication, the genome needs to get into the nucleus. So that's important. After penetration, the virus gets into the cell. And after encoding, the viral genome needs to get into the host nucleus. In the host nucleus, okay, the host DNA polymerase would make a complete double-stranded DNA to make the genome into complete double-stranded DNA. And from there, the virus would utilize the the RNA polymerase for transcription to make messenger RNA in different size. It can make messenger RNA like in 2.1, 2.4, 3.5 kb messenger RNA. Then the messenger RNA travel into the cytoplasm for translation to make the protein molecules. While for the replication, the HBV utilizes a special way. So here, the 3.5 kb messenger RNA is produced, transcribed. Then, to make the virion, this messenger RNA is packed into the virion, into the particle. Okay, the first step is packed in the RNA. Okay, we know that the genome is DNA. What happened? While it packed the messenger RNA, 3.5 kb RNA, it also packed the polymerase. The polymerase, we mentioned that it has a reverse transcriptase activity. So the polymerase would use the RNA as a template to make a negative sense strand DNA. So this is reverse transcription to make a DNA. Once the DNA is made, the polymerase has RNase H activity. RNase H activity means that it can degrade the RNA strand which complement with DNA so that it will degrade the RNA leaving only the DNA strand. After that, the polymerase can use a negative sense strand as a template to make positive strand DNA. So finally, you would have a partially double-stranded DNA genome. And when the HBV virion particle get mature, it can kill or release from the host cell. So this is roughly a simple life cycle of HBV. So that is why you should understand that how it utilizes the polymerase for its maturation. Okay, HBV. Okay, transmission of HBV is through three ways. The first one is blood transfusion and blood products. Like every time in the hospital when they give the patient some blood product or direct blood transfusion, they need to make sure that the blood is qualified, is free from HBV. And when you guys go to blood donation, they would make sure that your blood is clean. It does not contain any HBV virus in it. So that's now we know the existence of HBV. But before humans know the existence of HBV, actually there was a famous, interesting story happened in 1940s. You know, 1940s there was World War II. The U.S. armies were fighting in jungles, and in jungles there is a special disease called yellow fever. It's also a virus disease, and in America they want to make vaccine against yellow fever. At that time, the technique to make the vaccine utilize human serum in it, in the vaccine. So they went to the universities and colleges to ask the students to donate blood. And they picked the healthy students to donate blood and isolate the serum to make the yellow fever vaccine. But what happened is that after the soldiers received the yellow fever vaccine, some of them developed jaundice, which indicates the liver damage. And that's a time before we know the existence of HBV. But that's the first time that humans noticed the sign that in human blood, in human liver, there is some unknown virus which can cause damage. That happened in 1940s. And about 20 years later, scientists discovered the presence of surface antigen, HBV surface antigen. That was discovered in Australia. So at that time, they named it Australian antigen. So that's the first time that human ever identified the existence of HBV. And because of this work, the scientists got Nobel Prize later. So before the time of molecular biology, it's really hard to find something unknown. You know, you don't have PCR, you don't have sequencing. It's really hard to find a novel virus. So that's the first type of transmission. The second type is the vertical transmission from the positive mom to her newborn baby. Previously, we don't have any method to prevent such transmission. But nowadays, we can utilize the passive immunization. After the baby is born, we just administer some antibody to the newborn baby. And that is very effective to prevent the vertical transmission. The third one is a close contact. Here, the close contact means sexual contact. That's another way of HBV transmission. In China, it's a special culture and condition here. And because of the tradition of dining together in China, people share the food, share the dishes, and in China there are a lot of HBV infection. Well, at least previously, it was widely believed in China that if you share the dishes, it also causes HBV transmission. But I don't think there are a lot of evidence to support that. And even though in some families there are cluster of infection, a whole family got infected by HBV, but it's not clear what pathway, what transmission route is being used to cause that problem. I don't believe in the sharing dishes can transmit HBV virus. And during HBV infection, the virus infection does not directly damage the liver cell, but after infection, the immune response, such as the antibodies and cell-mediated immune responses, can recognize the infected cell and cause some autoimmune phenomena. So the immune response can target on the infected cell and kill the cell. So that's the major reason of liver damage. That contributes to the liver pathogenesis. This is HBV. Incubation of HBV is about 1 to 6 months. It can cause both acute infection and chronic infection. But we should keep in mind that HBV, especially for adults, even after exposure to HBV, most of the adults don't develop any disease. So that's subclinical. And even they suffer from hepatitis for some time. Most of the people infected by HBV can recover. And only a small number of the adults infected by HBV would develop into long-term chronic problem like chronic cirrhosis and liver cancer. And totally less than 1% of the total infected adults would develop liver cancer. So we should keep that in mind. Even though this total absolute number, the ratio is low, but if the absolute number is very high, like in a country in China with a big population, like 1.4 billion population, and it can cause a lot of problem. If a lot of people got infected, it still can be very expensive to treat those patients. So the best way to prevent HBV infection is that we should have some vaccine available. Okay, for laboratory diagnosis of HBV, nowadays we can use molecular methods to detect the presence of HBV DNA. We can use either nucleic acid hybridization or PCR to amplify the DNA directly, and that we can get the results within a couple of hours. But traditionally, in a hospital, this method is mature that we can run the serological test to detect the presence of antigen or antibody in your patient's serum. For example, if you find the surface antigen in the patient's blood, it means that the person is being infected, both acute or chronic. So if there is no infection, there shouldn't be any surface antigen in it. And if you detect the E antigen, which is processed from C antigen, that means a high level of HBV replication and infectivity. So that's the meaning of E antigen. And if you detect anti-C IgM here, anti-C antigen IgM, that's the antibody, which means the acute HBV infection, and this could be seen in flare of chronic hepatitis B. Well, if you detect anti-CIDG, it means that recovered or chronic HBV infection. Okay, and anti-surface antigen, it can be complicated. Okay, if a person previously infected by HBV, there should be anti-surface antigen. Okay, and if you are immunized, you should have this antibody, because in the HBV vaccine, we use the surface antigen as the vaccine. Okay. Well, anti-E antibody means low level of HBV replication and infectivity. This is a marker of treatment response. If you detect the anti-E antibody, it means that the infection is going down and or the treatment is working. Okay, so I'd like you guys to use this table to understand the serological test of HBV infection. Similarly, it's another table containing the similar information here. And just in case that in future when you are a doctor, if you have your patient's testing results, you can explain it to your patient. Okay, there is there are medicines available to treat HBV infection, such as we can use oral antivirus medicines. Usually they contain either nucleoside or nucleotide analogs. We learned that before. We can use them to inhibit HBV replication. And you can also use interferon alpha, okay, which may be effective for treating a chronic HBV infection. We explained the mechanism of interferon alpha and beta, why they can inhibit virus infection. While we can also use the passive immunization, that's HBV globin, the antibody, to block HBV infection. This is important for the newborn baby if their mom is positive. We give the baby the antibody the first week after born, and that can prevent the newborn baby newborn baby infection. Okay, we'll have a break. HBV infection was a serious problem in China a long time ago, and there are a lot of infections in China, but we didn't have effective vaccines available. Right now, we use the vaccine, the HPV vaccine. It's a subunit vaccine containing the surface antigen, the protein, as vaccine. And this vaccine is called a recombinant vaccine. So you clone the gene and then express it in either yeast or some microorganism, then purify the protein and make the vaccine. But previously, in China, there is no such vaccine available. By 1980s, the Chinese government want to get the vaccine from Western countries, and those companies in Western countries also want to introduce the vaccine to China, including the company called Merck. But the problem is that at that time, most of the Chinese people were poor, and the government couldn't provide free vaccine to everyone. And even though the company wants to sell the vaccine with a very low price, most of the Chinese people couldn't buy the vaccine. So there was a negotiation happened between the government and the company. And finally, the Merck company decided to provide China the HBV vaccine with a complete set of technique. And they provide a factory here in China with complete technique with a very low price, only like $7 million. It's very low, $7 million. And they also train the personnel working in the factory. They also allowed Chinese government to make more vaccine for the whole population. So that happened in 1980s. And by 1996, every child in China could get the HBV vaccine for free. And after that, the HBV infection rate is getting lower and lower in China. And right now, in the hospital, when every child is born, they would receive the first vaccine as HBV vaccine, so that the infection rate is very low now. But if you check the data, the global distribution of hepatitis B virus, you can still see that in China, there are still a lot of people who are positive, even though they are getting old. And you can also see on this map some countries with very deep color, that means high infection, such as Mongolia and some country in Africa, okay, which means that HBV infection is still a serious problem all over the world. And HBV is very infectious. The virus is relatively stable. And related to you guys will be a clinical worker, like a doctor or work in the hospital. You guys would have a higher risk of exposure. No matter you are a BDS student or MBBS student, you will have a higher chance of infection. So I would suggest you guys to check your immune status, have a serological test to see whether you have the antibody. And if not, you'd better receive a series of vaccine, HBV vaccine, so that you are protected when you work in the hospital. So this is the HBV. Okay, the third virus we want to introduce is HCV virus. It belongs to a family of Flaviviridae. Flaviviridae. Okay, sorry. This is the similar as the yellow fever virus. Belongs to the genus Hepatitis C virus. This genus only has one type of virus, Hep C virus. The genome of HCV is positive stranded RNA. Positive stranded RNA. Size of the genome, as I remember, is 9.4 kb. And HCV is not easy to study it because the virus does not grow in cell culture. So in the lab, you rely on cell culture or tissue culture a lot, but it does not replicate in cell culture. And if you use animal model, it only infects chimpanzee, the primate animal. So it's very expensive, and you would have a higher requirement of ethics approval. It's not easy to do experiments. And HCV, because of its single-stranded RNA virus, it can mutate fast. So HCV displays genomic diversity. And that is the reason why till now we don't have HCV vaccine. Because the genome is changing all the time. And it's not easy to design effective and long-lasting vaccine. So this is HCV. And HCV is envelope virus. See that? That's the envelope. This is the core, right? This is the HCV genome. Again, you don't have to remember the details. Just keep in mind that on the 5' end, it encodes the structural proteins, while on the 3' end, that's the non-structural proteins. Okay. And transmission of HCV is pretty the same as HBV, like blood products, and sexual contacts. So that's similar to HBV. Incubation period averages from 6 to 7 weeks. And the most new infections with HCV are subclinical. They don't develop symptoms. And those with symptoms some of them would develop chronic hepatitis. That include like 70 to 90%. And only a small portion of those chronic hepatitis patients would develop into cirrhosis. So that's the long-term liver damage. Okay, and HCV infection can also cause transformation. So HCV infection can cause the hepatocellular carcinoma, causing the liver cancer. So till now, we learned 3 viruses, and both B and C can cause liver cancer. Keep that in mind. And for diagnosis, we can use a serological test. That's a reliable method which have only recently become available. Okay, and HCV-specific IgG indicates exposure, not infectivity. You can also run the molecular methods, the PCR, to detect the HCV genome, HCV gene. Just keep that in mind. We don't have questions designed on these details. As I mentioned just now that we don't have vaccines available for HCV. We should reduce risks for HCV infection, like screening and test blood, to make sure that we can identify any possible of HCV virus source. We can screen the population and identify those persons carrying the virus, and test blood. For each blood donation, we should test whether it's HCV negative or not. And for treatment, the interferon alpha can be used, recombinant cytokine, which can inhibit HCV. Even though we don't have vaccines available, but we do have specific medicine available to treat HCV. This is just one of it to treat HCV infection. And I was told that the HCV vaccines are pretty effective, and it can control the virus pretty well, inhibit the virus pretty well. Okay, the next virus is HDV, hepatitis D virus. The D virus is about 36 nanometer in diameter. So that's bigger than the small particle of HBV, but smaller than the DIM particle. And it's encapsulated with HBV surface antigen. So this is interesting. In the virus of HBV, the surrounding protein is from HBV. The reason of this is that HBV is effective virus. Itself cannot infect human cell. So it always happens in a co-infection of HBV. And HBV virus is a helper virus. The specific antigen for HBV is the third antigen here. Okay, third antigen is encoded in HBV virus and is associated with the virus particle. So this is specific antigen. The genome of HBV is a single-stranded RNA genome. I guess you remember the definition of defective virus. Defective virus means that they themselves cannot replicate by themselves. They need a helper virus. This helper virus can be a relative or closely related virus or something not related. And this is for HBV. It requires HBV virus as a helper virus, but HBV virus is quite different from HBV because this is a DNA virus, while HBV is an RNA virus. And HBV infection only occurs in patients who are already infected with HBV. Okay, it can only cause co-infection. This is interesting, and it can make the disease even severe of HBV infected persons. Spread of HBV is again similar to HBV through either blood or blood products and body fluids like semen and vaginal secretion. So this is the similar way as HBV. It replicates and causes disease only in people with active HBV infection. This active HBV infection is important, okay, because it utilizes material from HBV replication. And the replication of HBV results in the cytotoxicity and liver damage. This is different from HBV, okay? You know, HBV infection, usually it does not cause direct cytotoxicity and liver damage. And the liver damage is mainly by the immune response. But HBV can cause this problem. It increases the severity of HBV infection, leading to the fulminant hepatitis. So that's HBV. And understanding the logic behind this, we should know the method how to prevent HBV infection. Okay, for diagnosis, we can detect the specific antigen, the delta antigen, and antibody, and or detect the HBV RNA, the molecular methods. To prevent HBV infection, the method is similar to prevent HBV, such as utilization of HBV vaccine. If you can prevent HBV, you can prevent HBV. So that's easy to understand. But vaccination by the HBV vaccine does not protect hepatitis B carriers from superinfection by HBV. So if a person is already carrying HBV virus, the HBV vaccine does not help anymore. So this is HBV. The last virus is hepatitis E virus. we want to compare hepatitis E virus with HAV, and HEV. So they share common characteristics. First, both of them are non-enveloped particles, naked viruses, and the size also similar. HEV is about 30 to 32 nanometer spherical, and genome is again also positive sense, single-stranded RNA. The genome size is also around 7 kb, 7.5 or 7.2 kb. Transmission of HEV is the same, it's fecal-oral route. And the symptoms and course of HEV disease are also similar to those of HAV disease. So basically, pretty close, but they're quite different. If you compare the sequence of the two viruses, they are different. This is HEV. We don't want to talk about more details on this. Okay, again, we come back to this table. So basically, we covered most of the information in this table. When you try to remember the characteristics of each virus, you want to compare them together, okay? Like which one contains the envelope, okay? Which virus can cause liver cancer, B and C, right? And like which virus we have vaccines available? A and B, and which virus can cause chronic disease? Yeah. Okay. So like the transmission route of each one. Okay. So you want to compare them and use this table to help you understand the material. It's not that difficult. Do you want to watch some video here?
If you have hepatitis A or B, your liver is inflamed because you have been infected with either the hepatitis A virus or the hepatitis B virus. Your liver is a soft, flexible organ that performs many important functions. The functional parts of your liver are called hepatic lobules. Your hepatic lobules filter all of the blood in your body. As the blood passes through, your hepatic lobules break down harmful substances. Remove bacteria and worn-out blood cells, and forms clotting factors that control bleeding. After a meal, your liver stores nutrients to provide your body with energy when needed. Your liver also makes a substance called bile. Your gallbladder stores bile and releases it into your small intestine to help digest fats in the food you eat. If you have hepatitis A, the virus entered your body when you were exposed to fecal matter from a person infected with the virus. You may have been exposed to the virus by eating contaminated food or water, contact with infected feces, such as during a diaper change, or having unprotected sex with a person infected with the virus. If you have hepatitis B, the virus entered your body when you were exposed to the blood or other body fluids from a person infected with the virus. This could have happened from sharing a drug syringe with an infected person. Other ways you may have been exposed to the virus include having sex with someone infected by the virus, sharing personal hygiene items such as razors or toothbrushes used by an infected person, direct contact with the blood or body fluids of an infected person, or when a mother passes it to her baby during birth. When either the hepatitis A or B virus enters your liver, it invades your liver cells and makes copies of itself. In response, your body sends immune cells to attack both the virus and liver cells infected with the virus. As a result, these liver cells become inflamed and then die. Over time, scar tissue forms around dead and infected liver cells, which prevents your liver from working properly. If you have a chronic hepatitis B infection, your liver contains a large amount of scar tissue called cirrhosis, which limits blood flow and results in permanent shrinking and hardening of your liver. If you have hepatitis A, your doctor won't prescribe any medical treatment because your immune cells will eventually find and destroy all of the hepatitis A viruses in your body. If you have hepatitis B, your immune system will usually remove all the hepatitis B viruses from your body. In some people with chronic hepatitis B, particularly children, their immune cells are unable to remove all of the hepatitis B viruses. If you have chronic hepatitis B and your immune system can't get rid of the virus completely, your doctor may prescribe antiviral medication. If you have a severe case of chronic hepatitis B, your doctor may recommend a liver transplant operation.
It's a very old video. It sounds like AI, but it's actually not. And they prefer such kind of style a long time ago. Okay, so you guys, anyone know your status of HPV immunization? You probably get the HPV vaccine when you were little, right? But do you have antibody in your serum? You don't know. The anti-surface antigen antibody. So that's the neutralizing antibody which can protect you. If you don't have that antibody, you are not protected. So right now you are okay, but just in case in a couple of years you'll go to study in the hospital, you'll have a higher chance to get in touch with patients and medical waste and body fluids. You want to get yourself protected. So if Zhengzhou University does not provide such service, you want to check your immune status, and you can receive a series of HPV vaccine to protect yourself. Okay, and you can get those vaccines from the local medical service center. It's not free, but it does not cost a lot. Okay? Now, if you work in Western countries, like in America, the hospital usually provides such service to give you the vaccine for free. But yeah, keep that in mind. Okay, so basically that's all for today. The hepatitis viruses. Okay, next time we'll talk about HIV. And unfortunately, next time is Thursday. It's Christmas Day. And for those of you guys who want to have a holiday, we'll have a holiday in the classroom. All right. Not everyone is having Christmas Day, but we have to come here. That's the last lecture. All right.